Hello, everybody. This is the recorded, and again, I'll have some other highlights probably just as I rethink it when we're in class, probably the latter part of Friday. And I'm going to do this one. So this is on D. So you folks at E, it's all the same thing. The tests are identical. And so what I do again, and I'm sorry if that one didn't open for you, but that's why I'm doing it again. I have effectively have the exam opened, and I basically recorded from this. So there's no visual. This is all audio. So if anything, there's 65 questions. They're all in Scantron. Nothing pet peeve. Bring mechanical number two pencils. Don't ask me. I won't have them. Drive me crazy. So, and understand I'm at home doing this and quiet because the dog is on nap. Nevertheless, but that never continues. So sometimes it comes in two parts. So the first chapter, there's probably 10 to 12 questions. I would say then maybe there's a good 30 questions from chapter 3 or from the bacterial related chapter, including some of the information from the eukaryotic chapter, which is chapter 4, and then maybe the other 20 questions or so, and these are ballpark numbers, but from the chapter on growth, which indicates basically if you're going to focus, your focus is on the bacteria anatomy and the distinction, particularly between bacteria and eukarya. So we start with, of course, Pasteur. Remember what he's about. His idea, everybody believed in spontaneous generation, like, magically the table comes alive, some vital force in the air. And he supported something called germ theory because he used a microscope and had seen particles. And his work after the whole wine industry thing and the development of what we now call pasteurization really was all about things that he observed with a microscope. So he was confident the spoilage organisms were that. And then he constructed or had the glass blowers construct those flasks, and then we reviewed that examination. So that's really the critical piece that's there. So again, Pasteur was a problem solver. And his colleague and maybe to some extent rival in Germany was Koch, was much more of the lab geek, as it were. So Pasteur was all about, he coined the word virus. He developed the first vaccine to treat rabies. He tried to look for other things in the early days of immunization. So that what the original Pasteur Institute was. So he was constantly looking for different diseases to work with, as opposed to, Koch, who basically developed Koch's postulates. We felt that you had to grow things in pure culture, establish that chain of evidence, first use of agar, really the isolation of all the things you're looking at in the lab. Koch looked at in the lab in 1900. So that's important. That's there. And a lot of what we talk about with bacteria, there's other people, Lister and Semmelweis and Edward Jenner. Jenner with smallpox even predates that. Reddy predating Jenner with the experiment with the flies. These are all noteworthy to remember because they were impactful. Lister, the father of what we call modern aseptic technique using diluted phenol solutions. Semmelweis with the childbirth fever known as puerperal sepsis, and the fact that he has established a link between effectively cleanliness and removing obvious germ-containing materials, dirt, debris, old dead body parts, and being able to obtain health for women. And that was, the doctors and medical students as opposed to the midwives. That's all very, very relevant. And really, bacteria have opened the whole world to us. If you want to think of it that way, just sort of, waxing poetic. I mean, they were there. And again, this is what science does. We looked at something you haven't seen before, really up close and personal in the age of molecular biology. And voila, bacteria are making these neat enzymes called restriction enzymes. And we do that in the genetics unit. And they chop up DNA. So it let us do DNA analysis. It let us do DNA fingerprints. It let us do paternity testing and ancestry and all that stuff. And because DNA is such a big molecule, but the restriction enzymes could chop up plasmids from one bacteria. And with molecular biology, we could splice the plasmids and insert them. And we found, amenable, friendly bacteria and also yeast. which are very simple organisms that were willingly to accept it. They're called recombinant organisms. And again, that's important. So what are genetic engineering is based on? DNA transfers from yeast to either E. coli, from DNA transfers from that have been created in the lab rather into E. coli or yeast. I spent a little bit of time on resurgent versus emergent. The big player in emergent surely is mutation. It's also to a certain extent the fact that, I mean, we've made advances and we can identify diseases now that we couldn't. And certainly we're going into areas and people are living in areas and they're contacting diseases that were probably there, but we in the more established world were unaware of as people began to reside in rainforests and what you might call jungle-like environments, or very, very cold or very, very warm environments that we hadn't really been to before. And there's a whole subset interesting ways. So we call those emergent, but I think mutation, and again, the focus point of antibiotic misusage are certainly important areas with regard to this. And then resurgent means it's something we got beat, we thought, but didn't. And again, really, tuberculosis is surely the most impressive of these. And because even without the use of a vaccine, just by testing and treating with antibiotics and chest x-rays and a lot of do's and don'ts, it was nearly eradicated in this country. But particularly the onset of immunological diseases and recognizing them, like HIV and then subsequently AIDS, immunosuppressive treatments that are used for people with severe immunological arthritis and MS and those kinds of things, as well as immunosuppressant drugs for transplants, which much of that's pioneered here in Pittsburgh at the Starzl Institute, increasing travel worldwide, vaccine laxity, aging populations. Those are the big players in resurgent diseases. And the vaccine laxity really applies to things like measles. And hopefully we want to get to a stage where they start to rely on things like polio. It's ridiculous. For successful vaccines have stood the test of time. They don't have to reformulate them with complications. Unfortunately, they've had to do a lot with COVID. So, I mean, that's the reality of it. Again, the antibiotic nugget is much more about things like mutations and the emergent aspect of diseases, because Staph aureus, as opposed to MRSA, when we get into our big unit on Staphylococcus, Staphylococcus was penicillin killed it reliably, and then all of a sudden now we've evolved it twice because of antibiotics. So I think that's important. Remember, the word unique plays a role in a lot of my questions, and typically I highlight or use boldface for these kinds of things on your exams that when you look at the paper copy. Unique in science means it's there but nowhere else. And surely the watchword for bacteria is peptidoglycan, because that's why it's such a wonderful target. That's unique. It's like chlorophyll being unique in the eukaryotic microbe world to algae. I know it's in plants, but they're macroscopic. So that's when it comes to reading questions. If you see the words microbe and chlorophyll, you don't even have to read the rest of the question. Most likely, the answer is algae. Do read the rest of the question and a note on that with test taking. I'll try to reinforce it when we take the test. Read the biggest single problem students have as they advance in the university or college environment with taking these kinds of tests is they don't read the questions. They answer the question. They don't read it thoroughly. They don't highlight things. They don't, because there's information in questions, not just for the question you're looking at, but for subsequent questions. So read the question thoroughly. You have the luxury of coming up and asking me a question. The reality is that I can't always give you an answer to that question. I might say I'm not able to give you any further information. I might be able to clarify it. There may be a misprint. I make mistakes all the time. So those are things that you may find a question where I've erred and put the same answer twice. Those are the kinds of things. Look, I don't make comments about my colleagues. If I make a mistake, I'm throwing out the question. Period. End of sentence. You get the points for my mistake. So I don't hold anybody accountable but myself. The other thing is trust your first impressions. There's when you study stuff, whether you know it or not, goes into your mind. if you write an answer down and when you go back and check, and do check the test, I don't want to check it because I'll change my answers. I always get in trouble. At the very least, you check it to see that you read it properly, made the mark in the appropriate place, didn't misread it, didn't put it in the wrong column or on the wrong number. You got to do all that. You only change answers. I'll buy that. If you mismarked it, misread it, or all of a sudden the light bulb went on in your head, which it does down the road because some other nugget in another question. And that's why it's so hard to make up exams that are challenging, because you're giving away the store in a lot of subsequent or other questions. So be aware of that. I know you don't see it this way at your stage of your careers, most of you. But the reality is, I always thought it was a great competition. It was me versus the professor or professors. That's just me. Yeah, make my day. Give me an exam that you can best me on. So that's yeah. Sometimes it's good to have an attitude like that, but that comes with time. So that's another thing. So, well, again, the word unique is important. So I do a lot of these. What differs from what? Bacteria versus archaea, prokarya versus eukarya. Read the question. For instance, bacteria and archaea, they're single cell with cell wall. They don't differ a whole lot, but peptidoglycan versus not an archaea, extreme environments versus not. Their cell walls are different and the environments are different, but they're roughly the same size wise and things like that. So I think that that's important. When we deal with, and we get a lot more, we have a whole chapter on viruses and highlight viruses, acellular forms, and prions in that chapter for our second exam. But for now, remember, viruses are unique. Anything in the living world is subject to a viral infection. E. coli and staph can get a cold just like you and I can get a cold, a viral infection. So all domains are vulnerable. And prions, as far as we know, are just proteins. So what do we know about them? Not a lot. They're not alive, okay? But they cause all sorts of diseases, chronic and otherwise, okay? They have a protein component, okay? They can infect, as far as we know, different species of different hosts. So all of those are part of it, okay? So understand the big distinction to me because the classification is non-living proteins. And if you want to think of them as being misfolded, that's fine. But viruses can do all that other stuff, chronic diseases, other hosts. They're very, very similar in what they can do and certainly can be fatal. But the distinction is viruses, DNA or RNA, never both, and most importantly, a protein coat, and they also use protein spikes, which we'll get to. But prions are proteins only. They're odd. And again, when we get to sterilization, which we are going to start in earnest uh, on Wednesday of this week, if you're listening to this today or before class tomorrow, that's what you got. The endospores. Spent a bunch of time on that. All about Pasteur and his work and the role the endospores played, and even in Koch's work. Again, for our purposes, very few bacteria, but particularly Bacillus species and Clostridia species. Those are the big players, and only under the right conditions. Yeah, okay. But the most important watchwords of this is Bacillus and Clostridia species, and depending on all the thing, not enough food to eat, whether there's oxygen or not oxygen, etc. They're effectively indestructible. They're in the spore stage, not the vegetative stage. Yes, the forespore becomes the endospore eventually. The peptidoglycan lays in it. Effectively, it's gram positive only that do this. Some of them are aerobic, some of them are anaerobic. And the coat is made of proteins additionally to really reinforce it. The dipicolinic acid component that basically is produced and attracts calcium and spits out water. All of these are part of it. Anatomically, there's limited numbers of forms that are there. If they appear circular or spherical microscopically, they're cocci. If they're rod-shaped, more or less, they're bacilli. And then there are forms that are spiral in nature. The spirochete is more of a they're curved forms certainly, but the spirochete is more of a corkscrew. So we basically have bacilli, cocci, and other forms, and most of those other forms have some degree of curvature. If you want to think of it that way. They're living things, bacteria. They all have a membrane. The definition of a membrane. phospholipid bilayer, and we're not talking about the LPS layer, even though it kind of applies to this. But the typical, when you talk about a plasma or cytoplasmic membrane, it's the one that contains the interior of the organism and a living thing. And it's a phospholipid bilayer with embedded proteins. And what defines it? Selective permeability. It keeps some stuff out, it leaves some stuff in. You want to go to the big game, you got to have a ticket. Okay. Basically, that's the way it is. It's a gatekeeper, and that's really important. I talked about going back to AP1 for many of you. We talk about what makes things move. And when you look at movement across membranes, again, like diffusion, facilitated diffusion, osmosis, active transport, vesicular transport, whether it involves energy or not. Diffusion is the most common. It doesn't involve energy. It derives its energy in the main player concentration gradient by far and away. I've emphasized that. That's really the other factors that play a role, sure. But what really is the driving force, concentration gradient. That's the most important. Okay. We look at some of the interior structures. Okay. In bacteria, they have ribosomes. They're 70S. They are located within the cytoplasm. They're not bound or attached to any membrane. They are within. When we talk about rough and smooth endoplasmic reticulum, that's a eukaryotic cell. So a lot of the questions are bacteria or read the question. So they're like any other ribosome. Protein synthesis is there. Okay. They have subunits of ribosomal RNA mixed with some proteins. That's what they are. A lot of my questions, which of the following are true, which of the following are false, which of these next statements are true or which of these are false or accept or more, you're going to see that. And you'll see I do a lot of sort of incorporate what's called a multiple, which means it'll be A, B, C, A and B, B and C, A, B and C, something along those lines. So be prepared. That's how you make standard objective questions more complicated. Why am I doing that? Because you see stuff like that on licensure exams, of which I have taken many. I've taken licensures now. I was licensed in 4 different states. I had standard board examinations, which qualify you to take licensure examinations. I was on 3 different boards. I'm blowing my own horn. Surgical and orthopedic and primary care board certifications. There's a lot of exams. Get used to it. So peptidoglycan, NAMs and NAGs. Okay, remember that. Only in bacteria. It's unique. Remember that. How is it cross-linked? Short amino acid chains called tetrapeptides. If it's gram-positive, they have the interbridges. There's teichoic acid, kind of anchors it because it doesn't just sit on top of the cell. It's got to be held there. And the big distinction is the amounts that we find in different bacterial organisms. Gram-positives and gram-negatives are not created equal. In gram-positives, you have large amount, 20 to 40 layers. In gram-negatives, 1 or 2 layers. So there's a major distinction when we look at that, which brings us, of course, to the gram-negative. So gram-positive, teichoic acids, thick layers, the peptidoglycan, it's inherently porous. It's a really useful target. It doesn't make one bacteria worse than the other, harder or easier to treat than the other, but they're all part of the picture. Whereas when you look at the gram-negative cell, because of that outer membrane, 2 things. One, it's got an extra piece of membrane surrounding peptidoglycan, which is encased in periplasm. And do remember the batting order from outside to inside or inside to outside. Outer membrane, peptidoglycan encased in periplasm, then the inner or cytoplasmic membrane, then the cytoplasm of the cell going in and reverse it going out. So sequence questions or those sort of anatomical location are important. I'm sure you've seen those on other tests that are there. So it's a neat way of putting structures. In my advanced physiology class, I'm using a sequence question it has to do with neurophysiology. So we use all of those things. Are part and parcel to how the exams are constructed. So when you look at the lipopolysaccharide layer, unique to gram-negatives, it's the outer portion of the outer membrane, and only gram-negatives. Toxins. Remember, all bacteria, mostly they do their dirty work by creating and secreting toxins because they spit them out. They're called exotoxins. Effectively, they exocytose them. The only one with an endotoxin is gram-negative, and inherently that lipid A of the lipopolysaccharide layer is an endotoxin. The chain that protrudes from it, there's that chain of sugars called the O-specific or polysaccharide or O-antigen, depending on the author or the source that you read, is an identifier. It doesn't contribute to its damage, but it lets us in the lab look at it and go, oh, this is, don't worry about this strain. If you get sick, it's minor versus, oh my God, it's O157H7, and we've got a contamination in the food supply that could kill a large amount of people and has, and will continue to do so. That's the difficulty. And really the difficulty, if you didn't know, and we talk about this a little down the road, in food preparation, you get a big factory and they're grinding up meat and somebody's not careful and they get some type of like intestinal contamination, fecal contamination, essentially from the meat they harvested and didn't treat it properly. All of a sudden, you got a food supply that, if they go to all the big, and most of the stores, if you didn't know it, get a lot of, one place prides them, they label them differently, and they'll go to Walmart, they'll go to Sam's, they'll go to Costco. It's all the same stuff. There aren't that many giant, cold packers and freezers who make this stuff. So be wary. Get an idea of that stuff. I mean, pay attention to these recalls because it could save you or somebody you love. So be aware of that. I hate to be preachy. So because there's membrane, the gram-negative inside and outside, what does that mean to you and I? It means, I'm going to plug back in my computer. It means we've got to be particularly careful, okay, with materials because we can't get through that outer membrane. So they have to have passageways called porin proteins, and sometimes we call them transport proteins, basically proteins with openings that allow non-fat soluble substances to get through. The sugar shell is important. The shell makes the slime layer, okay? It attracts other layers. It could be a multicellular association, but its formation has to do with that polysaccharide outer layer that we call a glycocalyx. So be aware of that. Again, a lot of my questions are prokaryote v eukaryote. And what that means is I'll say in a prokaryotic cell versus a eukaryotic cell, underline that. So in a prokaryotic cell, a flagella, we see flagella in both, but the flagella in a prokaryotic cell is made out of flagellin. In our cells, or in eukaryotic cells, it's made out of microtubules in that 9+2 arrangement, and they attach by a basal body. Not same. Our flagella, giant, compared to their flagella, a tenth of the size. Ours stick out like a hair. Theirs in the eukaryotic cell is encased in membrane. One of those appendages is made out of pilin, okay? Another relatively small protruding structure that's hollow because of the range the pilin proteins are made. And we classify that as a pilus, as the singular. The one we see the most of and is most relevant to our studies is called the F, or fertility or sex pilus. It's hollow and it's used for DNA transfer. It's not made of microtubules, it's made of pilin. And we'll see that a lot in our genetics unit. Plasmids, small circles of DNA with useful but non-essential. Again, the word essential is important because essential means it has to have it to survive. Plasmids can come and go based on the environment that microbes are in. So where do we find it? We find it in the cytoplasm. It may or may not attach to the nucleoid of the bacteria. Okay. I think that's important. It's a circle. And again, the DNA is roughly the same as ours. It's double-stranded DNA. The difference is the arrangement. Bacteria, it's circle, whether it's coiled or not. And ours is basically in linear segments. We have pairs of strands, good-sized strands of chromosomes that are there. It replicates along with the cell ordinarily, whether it's attached to the the DNA of the bacteria separately in the nucleoid, or if it is not. When you talk about ribosomes, that's another big difference, 70S versus 80S. The typical ribosome in you and I is an 80S unless it's inside of a mitochondria, that's a 70S. Or inside of an algae, that's a 70S within the chlorophyll portion of the chloroplast portion. That's there. So there's distinctions that you have to be, and that's something to look at. The same thing, if you want to go back about endospores. Remember, review that video. It's there for you. It's easy. What happens? It basically separates in such a way that it undergoes fission, but asymmetrically. So you have the DNA is separated, but the mother cell DNA has a vast amount of cytoplasm. The forespore does not. The mother cell's membrane engulfs and creates a double membrane where peptidoglycan is laid down. And when that is accomplished, the mother cell's DNA degrade. When does it happen? With decreased amounts of carbon and nitrogen and or water that are there. Peptidoglycan is laid down, and then it involves a rather substantial amount of time until the creation of the protein code and the exosporium, depending on what it is. Okay, it has to lose water, it has to attract calcium. All those are part of it. But the big player here is the idea that it's an adverse condition to thrive, and that you need to remember. All eukaryotic cells have sterols, animal cells have cholesterol, fungal have ergosterol or ergosterol. That's the big player to stabilize the membrane. Okay, animal cells don't have cell walls at all. That's our big distinction between plants, if you want to look at it. And there's others when you look at the individual cell types. And even though years ago we didn't think there was a cytoskeleton, there is a cytoskeleton in bacterial or prokaryotic cells. It's tiny filamentous fibers, but they are not necessarily, maybe there are tiny bits of actin in them potentially, but there's some other fiber-like material. They're not anywhere like microtubules or intermediate fibers or the fibrous actin that you see in muscle physiology, with actin and myosin sliding over each other. So prokaryotic cells, much less organization, much less advancement than eukaryotic cells. The old question, where's the ribosomal RNA? Everybody's test question. Ribosomal RNA is made in the nucleolus. None of those we find in bacteria. What do mitochondria and chloroplasts do in eukaryotic cells? Energy centers, energy transformations. Pure and simple, nothing special. They're only in eukarya, and that's what they do. Chloroplasts use light energy. Mitochondria effectively use organic materials for energy for the most part for our types of cells. And yeah, that's the big player. Facultative versus fastidious. Facultative means they have the capability of growing in a variety of conditions. So when you look at the facultative anaerobes, like you look in the lab, they grow in the presence of oxygen, but they can do energy transformations in absence of oxygen by fermentation. So they're not affected by the absence of oxygen the way you and I would be. That's facultative. They have the faculty or the capability to do that. Certain characteristics we associate with eukaryotic organisms are being saprobes. Bacteria do this ordinarily. Bacteria decompose dead and dying things. Fungi, yeast, and mold do the same thing. But it's a neat process. You don't have to know the name. It's called exodigestion. They secrete, once they invade, they secrete enzymes that break it down. But it's basically the roadkill approach, how it deteriorates over time. So by that, a saprobe effectively is a decomposer. And decomposers, by definition, live on dead and dying material. So again, remember the batting order of stuff going in, and the gram-negative from the exterior to the interior to the exterior, depending on how I phrase it. Remember the structure of the gram-positive cell, the fact that it doesn't have an outer membrane, has a lot of porosity and those multiple layers of peptidoglycan. It's got teichoic acids, Okay, the polymers are carbs with nitrogen in them. That's the glycan. That means sugar with nitrogen. And and because that porosity, small, comparatively water soluble things can get through. Big things can't get through. So water, electrolytes, that type of stuff can get through. The other stuff, it's going to have a difficult time to get through. Big stuff, complex carbohydrates, nucleotides, proteins, we classify what we call macromolecules, whatever their solubility. The big temperature levels, mesophiles, psychrotrophs, refrigerator, thermophiles, hot tubs, hot springs, that type of stuff. Roughly know the temperatures. Remember, mesophiles, 25 to 45, it's in degrees Celsius. And let's say the thermophiles, the ones like a little warmer, roughly 45 to 70. There's always some overlap that's there. What happens in the fridge? We reduce food spoilage. How? All about how rapidly. It's all about the metabolic rate of the organisms. The organisms didn't die. Cold doesn't kill them. Cold slows them down. Freezing them doesn't kill them. It basically maintains them until they warm back up. That's part of it. So there's no irreversible process that goes on. We don't see freezer organisms are rare indeed, psychrophiles. So we can't see that they necessarily grow. They just exist in that type of environment for the most part. And remember, it's in the refrigerator, not the freezer, so it's different. What makes an anaerobe obligate? Most importantly, it can't use oxygen, but it's killed by oxygen. It doesn't have the means to deal with toxic aspects of oxygen, those toxic derivatives. So it can't detoxify oxygen. It's rapidly destroyed by oxygen. It doesn't have those enzymes, catalase or superoxide dismutase. There's not some, there's none. And yeah, when do we see Clostridia is the poster child for that, in the sense that they find them in the soil, and really that's where the endospores end up. It's a sort of a secure environment that they can be found in. So you need to remember all of those things. The microaerophiles, remember, they require reduced, so slightly devitalized tissue. Certain areas in us where we have a lower amount of oxygen, so they certainly affect us. And again, the role they play in ulcers and with the flesh-eating bacteria, you need to note, and we cover that again down the road. Okay, so they only can function in reduced oxygen environments. Sitting out in the atmosphere here today, outside your house or outside your dorm, not happening. That are there. The toxic derivatives of oxygen, we talked about a lot. Superoxide dismutase, particularly, that happens during the normal use of oxygen. They're a big player in cancer. Modern cancer and aging theory is all about these free radicals. We have different things called, and we talk about that a little bit down the road, more or less in viruses. We have initiators and promoters, things that start the deterioration process and those that make it continue. And some are both, like cigarette smoking is both. Okay. Neutrophiles or neutrophils are different. Neutrophiles are the bacteria. Our textbook is 6 to 8. Other textbooks are 5 to 8. As long as it's in that range, that's where they flourish. How do we basically destroy things? And it's based on how they grow, as you'll see in the next chapter. But noteworthy, and we saw a slide about this, when you place them in isotonic media, nothing happens to your cell or a bacterial cell. If we place them in hypotonic media, our cells would burst. Okay, that's a big problem. They would undergo plasmolysis. The plasma membrane would burst. On the other hand, bacterial cells would not, because they have a cell wall to reinforce it. In a salted environment, sucking water out, now that would lead to bacterial cells would undergo plasmolysis. So it's different. Our cells, too much water without the security of a cell wall, it bursts, plasma membrane lysis. Their cells, bacteria, The phenomenon is reversed because remember, it's an envelope. It pulls the wall, and the membrane pulls away from the wall, and then it bursts. That's really significant. Okay. The cell wall can only protect it when there's too much, but not enough. Again, heterotrophs eat everything, use that organic materials for their carbon sources, use it for their energy resources, where autotrophs, self-growth, sunlight and atmospheric carbon dioxide, they fix carbon. Again, facultative versus fastidious. What's fastidious? Many different growth factors, whether it needs special environments or special chemicals or special materials. We talked a little bit about Neisseria and Streptococcus fall into this. Remember CHONPS for bacteria. So carbon, hydrogen, oxygen, nitrogen, plus phosphorus, plus sulfur. A little different than us. With phosphorus the same, but we have calcium. Don't forget run, tumble, run. Basically, it's attracting or repelling, whether it's, and most common is chemical, certainly, but there's magnetic, there's light, there's air, there's thermal. The next set of questions refer to, and it's a multiple, it's my version of matching. Get used to my version of matching, which means you can use once, twice, or not at all. Sorry. So this is all about the bacterial growth curve. 6 questions. Know it well. Lag phase. Getting ready to reproduce. It's accumulating nutrients and materials so it can, because it can be in this active replicative process, okay, that we call binary fission. So that's what it does. It's making essential macromolecules needed for growth. Log phase, exponential growth. What does it do? Okay, what's noteworthy about that? It's most susceptible there because all it's doing is growing, it's gobbling up anything in its environment. Anything toxic in the environment will kill it. Also, primary metabolites, which are useful, that we talked about before, they're produced. As the conditions become adverse, as the nutrients and the waste nutrients disappear and the waste products accumulate, we'll move into stationary phase where there's no more nutrients in the media in this closed system, and waste products are creating a ton of adversity, lack of carbon, lack of nitrogen, lack of effective water. What happens? Endospores, if they can make them, they do so, even though they start in late log phase, but they're a hallmark of stationary phase. And then your waste products of the bacteria who can survive, because it's about 50-50, those that are living and those that are dying, those that can survive, okay, make a secondary metabolite. That's typically where we harvest antibiotics, if they have the capability to do so. And again, just like endospores, they all don't do that. And then in death phase, they're more dying than are living. And it's linear in a sense of it's a set rate, but not nearly as rapid as log phase, which is typically very rapid. And pretty much the bulk of the remaining questions are true, false. To be true, it has to be absolutely true. If it's and, it's got to be, both things have to be true. It's not this is true and this is not. If anything makes it false, it's false. If it doesn't apply, it's false. To true, it has to be absolutely true. That's how you understand true, false questions. So things like that. Okay. Gram positives and gram negatives are in all the temperature categories. There's nothing unusual about that. Certainly in the major categories that affect us, the psychrotrophs, the thermophiles, and the mesophiles, gram negatives and gram positives exist in both those worlds. They all replicate by binary fission. There's some unique ones that we really don't study that do play a role in antibiotics, but effectively it's binary fission. It just looks a little bit different. The distinction between, again, the biggest thing is no membrane-bound organelles. So ribosomes, by definition, are free in bacteria and archaea. They are either free or bound because they attach to a membrane in an internal membrane in eukaryotic cells. All living things have that phospholipid bilayer. Nothing unusual about that. Endospores. Remember, this always confuses. Endospores are normally, they only form in the adverse stage. The endospore or spore stage is under adverse condition. Normally, they are vegetative cells. Vegetating for bacteria and for plants is healthy, reproducing activity. Okay, and it's all about favorable environment. We see plasmids all the time. And part of it, I didn't comment on this, but I mentioned it in passing. Emergent diseases. MRSA is emergent because it mutated. COVID, which is part of what's called SARS, okay, severe acute respiratory syndrome. Those are all from coronaviruses. I did make this point. It's important. I'll reiterate it in class. So COVID-19's name is SARS-CoV-2. That was the original COVID-19 virus, because the first time we saw SARS was in an airplane coming from Japan to Canada back in around 2001, 2002. So, and there's been more than that. There's also something called MERS, Middle East Respiratory Syndrome, which we didn't discover so much as it occurred, particularly in our military when they were over in Desert Storm. Okay. All bacteria produce exotoxin. Doesn't matter if they're gram negative or gram positive. Only gram negative is endotoxin. If you're an aerobic cell, you got to have a peroxisome. Again, understand that prokaryotic cells, a single circle of chromosomes. It's pretty big, just supercoiled. Eukaryotic cells, pairs of chromosomes that are there. And again, the distinction between internal structures like cilia and flagella and eukaryotic cells sheathed in membrane versus in prokarya, where fimbriae, pili, and flagella protrude like tiny hairs. Okay, and don't forget generation times. The typical time is around 20 to 30 minutes for most bacteria, with some notable exceptions. And ladies and gentlemen, that's it. My computer's going to die, but that is everything for us. So see you on Wednesday if you're listening to this today, which is Tuesday the 6th. Bye-bye.